Okay, well, welcome to the first afternoon section. We're going to have now two plenary talks. The first plenary talk will be given by Professor Moyses Nussenzveig. Professor Moyses Nussenzveig is a very well-known scientist in Brazil and abroad. He is one of the pioneers in light scattering and some other aspects of quantum optics and many other topics related to optics. He was a professor in Rochester, and then he came to Brazil. He started in Brazil, moved to the United States, came back to Brazil, and now he is a retired professor from the Federal University of Rio de Janeiro. And he's been a very important driving force for optics in our country. So I'm very pleased that he's going to give us a nice talk about light and life, which is very appropriate for this meeting and for this year, which is the International Year of Light. So Professor Moyses, thank you for accepting, and you have now your time for your plenary. Thank you.
Thank you very much, Vanderlei, and thank you for the invitation. And as you can see, Bohr, about 80 or more years ago, gave a lecture with this title at the International Congress on Light Therapy. So this is a very good place to talk about this topic. And what I'm going to do is to revisit his talk and find out whether 80 years later it still makes sense, what has changed since then. And his main question was, is quantum mechanics relevant to biology? So let me start saying that as a physicist, I had to acquire some biological insight, and the most important one is related to Darwin, who plays the same role in biology that Newton does in physics. And the geneticist Theodosius Dobzhansky said, nothing in biology makes sense except in the light of evolution. Now, this is from Bohr's paper. I'm putting these framed. He started by saying that the absorption of a single photon in the eye is sufficient for a sight impression, so the sensitiveness of the eye has reached the limit set by the atomic character of the light processes. This is not quite true because although the eye does detect one photon, it takes about 3 to 5 photons for a visual sensation. But similarly, the threshold for hearing is an air displacement in the ear of about 1/10 of an atomic diameter. So how come that we are at the limits of sensitivity in both cases? Well, here is where Darwin makes this statement is important, because there are obvious evolutionary advantages for our predecessors to be able to early detect a predator in the jungle. Another important thing which Bohr was aware of is that in biology you have to take teleological arguments into account, because differently from physics, living organisms have a program which is the self-generation of individuals, okay, and their growth. So let me give a very brief timeline of some very important contributions. One question is life consistent with thermodynamics? After all, organisms increase their order in time, and so there's an apparent contradiction with the second law. However, this is the famous set of lectures by Schrödinger called What is Life?, in which he mentions that what living organisms feed on is neither matter nor energy, but negative entropy. And in fact, Boltzmann in 1875 already said it, that it's the struggle for negative entropy which is crucial, and that becomes available through the transition of energy from the hot sun to the cold earth. So we feed on order, which is created by the sun. And in this book and his lectures, he tried also to explain genetic stability and inheritance by quantum mechanics, by modeling the gene, of which little was known at the time, as something that quantum mechanics says has stability, like a crystal. He said it's an aperiodic crystal. You could think of it as a biopolymer in today's language. In fact, he acknowledges the influence of Delbruck, and Delbruck in turn decided to move from physics to biology because of Bohr's lecture in 1932. A crucial contribution also was given by Alan Turing in 1952. He was studying, trying to understand the riddle of morphogenesis, how from just primordial cells there is a whole organism that's built up. And for that, he assumed that there must be exist substances that he called morphogens that diffuse through tissue, chemically react to create patterns. And that's part of the story, but a very important part. And for describing this, he introduced reaction diffusion equations, which are generalization of Fokker-Planck equations. Now, everybody knows about the next step. That was in 1953, when on February 28 at 12 noon, Francis Crick entered the Eagle pub in Cambridge and proclaimed, "We have discovered the secret of life," with a typical physicist modesty. Well, was it really? Certainly it was a very fundamental discovery. It provided the mechanism of gene replication, and it was followed by the unraveling of the universal genetic code. And for instance, the role of transfer RNA was guessed, hypothesized by Crick. Now, of a comparable importance was the discovery of the regulation of gene transcription by François Jacob and Monod in 1961. And another less well-known, very crucial discovery was by Peter Mitchell in 1966, what he called the chemiosmotic hypothesis, which is again a universal mechanism for energy production in cells. And Leslie Orgel has called it the most counterintuitive idea in biology since Darwin. So let me talk a little bit about the origin of life. I strongly recommend this book by Nick Lane. But the origin of the simplest living thing, which is a cell, which is already a very complex system, poses a typical chicken or egg problem. This cycle that DNA, which contains information, gives rise to messenger RNA, and from there proteins, is all catalyzed by these proteins. So it's, which came first here? Well, there are really two problems for the origin of life. One is, what is the chemical origin? How did the ingredients arise? And the other is the energetic origin. What is the mechanism of energy generation? Now, the idea of the mRNA world is a very important step in this. The idea is that it was discovered that RNA not only stores information, but can also act as a catalyst, ribozymes, and as a regulator. And more recently, RNA interference, a very important mechanism for regulation. So this helps a lot. Now, of course, in the origin of life is hypothetical, but the most accepted hypothesis for the energetic origin and part of the chemical origin is that life arose at deep ocean hydrothermal vents, alkaline hydrothermal vents, which are kinds of chimneys which can be nowadays, one of the most famous ones was discovered only in 2000. It's called the Lost City Hydrothermal Field. And these have a flow of very hot kind of chimneys arising from combination of volcanic and plate tectonic origin. And if you look at their microstructure, you see that they are formed by micropores, which are very small. Their typical size is the size of a cell. And the walls are lined with catalytic iron and sulfur, and these behave as natural flow reactors that are powered both by thermal and electrochemical gradients. This one in particular has lots of bacteria and archaebacteria in it. Now, within these pores, hydrogen and CO2 accumulate, react, and in the end produce acetyl phosphate, which is an analog of ATP and therefore can be seen as a precursor of the RNA world. Now, very crucial step was going from prokaryotes to eukaryotes. And about the chemical origin of life, a very important paper was published just a couple of months ago in Nature Chemistry, showing that precursors for all the building blocks in the chemistry of life, nucleic acids, amino acids, and lipids, could have arisen at about the same time from HCN and SO4, where the HCN is deposited by comets, and this with the help of ultraviolet light. The first remnants of terrestrial life are traces of prokaryotes dated about 3.8 billion years ago, and you can see here a fossil of 3.5 billion years old compared to a modern cyanobacteria. You can hardly tell the difference. And these prokaryotes have two branches, bacteria, or eubacteria as they're now called, and archaea, which initially were classified as bacteria but were then recognized as a separate domain of life, quite different from ordinary bacteria in several significant aspects. Now, both together are still the dominant life form on Earth in terms of biomass. Well, they are supposed to have arisen from a common ancestor, denominated LUCA, last universal common ancestor, which has the two universal features of all life, the universal genetic code and the use of chemiosmosis to generate energy. What is chemiosmosis? It's employing a proton gradient obtained by pumping across a membrane to generate an EMF. And this is used, for instance, for the synthesis of ATP, which is the universal currency of energy in life. And this gradient is about typically 150 millivolts, but over a 5 nanometer membrane, that is non-negligible. It's 30 million volts per meter. Okay, now the emergence of eukaryotic cells is considered the most important event in the evolution of complex life. And it is now generally accepted that the eukaryotes originated from a unique encounter between bacteria and archaea, that archaea swallowing a bacteria, and that established an endosymbiotic relationship, giving rise to mitochondria, which are the powerhouse of all eukaryotic cells, and preserve many prokaryotic features. Now, still more recently, only about 2 weeks ago, a very important missing link in this evolution was discovered, called Loki. It's a type of archaea which already has several features of eukaryotes, and also discovered in a hydrothermal vent. Now, the presence of independent power sources within the cells permitted the genome to have a huge expansion, and that is what gave rise to animals and vegetables and all the other eukaryotes. And a further endosymbiosis with cyanobacteria gave rise to chloroplasts and the mechanism of photosynthesis. Okay, sunlight plays a crucial role in life. Starting about 2.5 billion years ago, the cyanobacteria oxygenated the atmosphere, led to oxygenic photosynthesis, which is represented in detail here, but I won't have time to discuss it, and cellular respiration, which is sort of the reverse mechanism. And through those, all life on Earth is nowadays maintained by sunlight. The photosynthesis produces nutrients and returns oxygen to the atmosphere. The cellular respiration breaks down these nutrients, restores this crucial proton gradient for the generation of ATP. And both the chloroplasts and mitochondria employ chemiosmosis, which very plausibly could have arisen in hydrothermal vents, which are alkaline and bathed by an acidic ocean. Nowadays, we have the average Earth surface temperature at a comfortable level, thanks to the greenhouse effect. Okay. Now, a question that Bohr could have asked, is quantum mechanics relevant to biology? Well, he commented that it offers a clue to the intrinsic stability of atoms. And that's absolutely true. It's even a theorem that matter is stable because of quantum mechanics together with the Pauli principle. And the structure of matter as well as chemical processes is explained by quantum mechanics. However, are the biological interactions, do they require a quantum mechanical explanation like covalent bonds? The answer is no. Typically, they are much weaker. They are van der Waals-London hydrogen bonds, ionic or hydrophobic. And this makes all sense because of Darwin, because it is required for flexibility. Proteins fold, and all this requires this flexibility. It would not be possible if it were strong chemical binding. Does quantum coherence play a role? Well, I don't have time to go through this, but the answer, as far as I can tell, is no, it does not. And that's consistent with evolution also, because quantum coherence is something very fragile, fast destroyed by environmental noise. Now, Bohr also discussed the brain and mind, saying the interaction between the measuring instrument and the object exhibits a close analogy to the fact that the mental content is altered when you concentrate attention on it. Well, the mature human brain is the most complex system that we know in the universe. It has almost 90 billion neurons, each connected to about 10,000 others and another number of non-neuronal cells. So the number of connections is larger than the number of stars in our galaxy. Each neuron itself has a complex structure. And the signals, this works, the whole system, by highly parallel as well as distributed operation. The signals are processed and reprocessed in both directions among neuron layers. And they are organized in modules that are interconnected. And they change all the time, responding to the environment. A complex system involves a large driven conglomerate of nonlinear interacting elements, and one characteristic property is self-organized criticality, spontaneous appearance without tuning a control parameter of bifurcations which have long-range space-temporal autocorrelations that are scale-free decay with fractal exponents, and they give rise to unanticipated collective emergent properties and bursts of activity known as avalanches. Now, a very interesting paper quoted here shows by modeling that the resting brain has precisely this situation of sitting at one of these critical states. It's tuned to a critical state. And again, this makes sense from the point of view of evolution because it allows maintaining enough order for coherent functioning, but also a degree of disorder and flexibility to respond to a varying environment. How about consciousness? Bohr said, recognition of the limitation of mechanical concepts in atomic physics would seem suited to conciliate the apparently contrasting viewpoints of physiology and psychology. Well, Francis Crick, who at the end of his career turned to brain research, wrote this book, The Astonishing Hypothesis. And he defines it this way: you, your joys and sorrows, your memories and ambitions, your sense of identity and free will are no more than the behavior of a vast assembly of nerve cells and their associated molecules. As Lewis Carroll's Alice might have phrased it, you're nothing but a pack of neurons. Well, how reliable is consciousness? There are several illusions. For instance, we all have a blind spot in the visual field of which we are not aware because the brain fills it up. And phantom limbs that are felt by many amputees can be cured, as shown by Ramachandran, by showing in a kind of mirror box the normal side, the arm, together with the virtual one, and the illusion this creates sort of cures a kind of virtual amputation of the phantom limb. And one can even have the illusion of body swapping, again, an effect of the brain. It is something which all scientists have experienced, I am sure, and as is related by Poincaré on how he discovered automorphic functions, that this was done at a moment that he was not at all thinking about this. He was just going on a bus, and suddenly he understood something which was very crucial for mathematics, as a kind of sudden illumination, of kind of eureka. Okay, how about free will? Bohr said, the freedom of the will is a feature of conscious life that evades a causal mechanical description and resists physical analysis. Well, this is not true any longer. The onset of cerebral activity in one famous experiment in 1983, preceded by a few hundred milliseconds, the time that the person became conscious of the intention to act. And the conclusion then was that the cerebral initiation of a freely voluntary act can begin unconsciously before any awareness of the decision to act. In fact, papers have shown that apparently free choices can be predicted in advance. And a very important application, medical application, was published in Science just 2 days ago. Okay. So, what is an important contribution is this global neuronal workspace model described in this book by DIN. And again, it shows that consciousness lags the external world. Most of the brain's operations are unconscious, and consciousness arises typically around 300 milliseconds after an unconscious decision has already been made. The prevailing view among neuroscientists, both consciousness and free will are emergent properties of our brain. Now, I don't have much time to talk about light as a tool for biology, although most of this meeting is related to that. But in terms of fundamental biology, a fundamental contribution was given by Arthur Ashkin with the invention of optical tweezers. I assume that most of the audience is familiar with that. And one important step has now just recently been achieved at our lab. We have managed to complete a long series of papers aiming at the absolute calibration of optical tweezers, and we did manage that. That's published just last year. Without any fitting parameters, it explains the force produced by tweezers in all experimental conditions, and we're currently applying it to measure Casimir forces, which are very weak. They are femtonewton forces. A very important application of optical tweezers is single molecule cell biology. And here Bohr fell into a trap. People often attribute to him this saying, prediction is very difficult, especially about the future. Well, this applies to Bohr because he said in 1932, we should doubtless kill an animal if we try to carry an investigation of its organs so far that we could tell the part played by single atoms in vital functions. Now, nowadays there is a magazine called Single Molecule Cell Biology, and a lot has been done also to explain how proteins work. Here, the basic idea goes back to Monod in his book, Chance and Necessity. He states, proteins are Maxwell demons. I don't have time to go into details, but yes, Maxwell demons are really basic to understanding how proteins and motor proteins in particular work. And this has given rise to the automatic Maxwell demon popularized by Feynman, was called the Brownian ratchet. And the conclusion, again, I don't have time to go into the details, is that motor proteins work by rectifying Brownian fluctuations. And life as we know it would not exist without Brownian fluctuations. Okay, I have very little time to discuss details about these motors that were discovered using optical tweezers, but let me just mention a couple of things. First, that one can measure, this is called mechanobiology, one can produce tethers, extracting them from cells by using optical tweezers. We do this a lot in our lab. And you can measure the elastic properties of cell membranes and show that they have an effect on the function that the cell exerts. Also, a very interesting feature called tunneling nanotubes can be exploited with optical tweezers. These are membrane nanotubes that cells extend spontaneously to connect to distant cells. I don't have time to give the details, but let me just mention that they have very important features. They act in cell-to-cell communication like a kind of parallel nervous system. Okay, and they're apparently very old in terms, have an ancient evolutionary origin. They exist in vivo, and they are also, from recent work, involved in cancer cell propagation. So, summarizing what is in Monod's book, chaos and order are fundamental ingredients to understand life. They play complementary roles. Order is in the genome, in the molecules on which we feed produced by sunlight, and in these marvelous molecular machines, the proteins. Chaos in the fluctuations affects the genome, random mutations, is the source of Brownian motion, explaining how proteins behave as maxodemons. It played a role in the origin of eukaryotes and also very important in the diversity of the immune system. Even the thermal ratchets, in a way, are Darwin's motors. They make natural selections of favorable Brownian fluctuations. So more than 8 years ago, Bohr could not have foreseen the amazing technological advances of biology since that time. But his paper is an example of the fact that finding the right answers is less important than asking the right questions. And as he stated when revisiting his paper, life will always be a wonder. What changes is the balance between the feeling of wonder and the courage to try to understand it. So this is the beautiful epitaph of Stevin, showing that the explanation of an inclined plane is, as he put it here, Wunder and Heilwunder. It's a wonder, but not a miracle, that applies both to life and to light. I have to acknowledge the work of our team at our optical tweezers laboratory. Thank you very much for your attention.
Hey, thank you, Moisés. It's a lot to be saying in 30 minutes, right? All life and all light. So we have time for one question, if you have a good one. Okay, well, I have one then. How the physicists, atomic physicists mainly, are taking serious some of the things that Bohr said long ago. So how many atomic physicists are really trying to make the connection and putting quantum mechanics inside the life and everything, even though we know that it's there already?
Not many of them, unfortunately, but I didn't have time to show one beautiful example, and that has all to do with this Congress here. This paper published 2 days ago was a new technique for a brain-machine interface for a tetraplegic. This even appeared in the national news here. And the basic idea is that the brain takes a decision long before the person becomes aware of it. So that now implanting electrodes in the part of the brain where the decision originates, you can instantly produce the motion that the person is planning to do before the person becomes aware that he wants to do that.
Okay. So let's thank again Moisés for the very nice talk.